So welcome everyone, and thank you for being here this morning or this afternoon, according to your location. I'm Alessandra Ferrajoli. I will be chairing for the third time the Paola Campese Award. First of all, a few words regarding ISSNAF, that, as you know, is a very important association for us that bridges the scientific community between the United States and Italy, in particular all North America. And is represented by Dr. Cerruti here today. Regarding the Campese Award, this is an award that has been established by Dr. Vito Campese in honor of his daughter Paola, that unfortunately died from acute myeloid leukemia at a very young age. So in order to honor her memory, she was a very brave, intelligent, and beautiful woman. He decided to establish this award. Moving into the event of today, first of all, congratulations to all the three finalists. Only one of you will be selected for the award. However, you should be very proud of being a finalist and note that in your CV, because being a finalist is already a great honor. I will also take the opportunity to thank my two co-chair and reviewer that are here today for the evaluation, Dr. Marco Ruella and Dr. Paolo Strati. And there couldn't be a more appropriate team considering their expertise in CAR-T, that is the subject of two of the three presentations today. Instead, I represent the more pure leukemia field. Without any further ado, I think we can proceed with the presentation. That will be in alphabetic order. Each of you is going to be given 15 minutes for the presentation, then 5 minutes for questions. I would like to make sure that you consider this setting very familiar, and any question that will be asked will be just for further clarification, and maybe also to provide some input on your research that has been really impressive. So thank you for participating. Thank you for sharing your achievement. And we'll be starting with the first presenter, that is Dr. Nicoletta 𝐏𝐚𝐅. And her presentation title is Systemic Identification of Minor Histocompatibility Antigens Predict Transplant Outcomes. Nicoletta, you can start.
Thank you. So first I have the list to share. Allow to share screen. Okay, I'll share my entire screen and then. Do you see my screen?
Yes, we do.
Yes, we do.
Perfect.
Okay, perfect. So first of all, thanks for selecting me. And I'm happy to share the results we had. This is basically the activity of my postdoctoral time in Boston. And I work, and I've always worked in transplant, which is always kind of a difficult field to describe to the outsiders, because what we do and what we keep doing, because it's probably one of the few potentially curative options for many patients with leukemia, in particular with acute leukemias and myelodysplastic syndromes, is that we transplant the hematopoietic compartment of a donor in a patient, and this is kind of a sort of a black box in which how we select the donor is very rudimentary, because we use HLA typing, age and gender of the donor, and not so much else. And then what we do is that we hope for the best and for the immune system of the donor to be able to exert what we call the graft versus leukemia effect, which is the beneficial effect and the donor T cells that recognize as known self any potential residual disease, so thereby ensuring long-term control against disease. But at the same time, those donor T cells can recognize also molecular determinants outside of the hematopoietic system and virtually in any donor, in any organ of the recipient. And that is what we call the graft versus host disease. And I mean, the last I would say 50 decades, have been devoted to try to understand and how to disentangle the GVL from the GVHD. And to get here, basically, the most important thing and that drives the effect of the transplant is alloreactivity. So this recognition as known self by the donor of the patient. And what is recognized as broad categories can be either inside the HLA, because we know that HLA molecules are very potently immunogenic, and that is what happens when you have donors that are mismatched at the HLA loci, which could be unrelated donors from the registry that do not share a full HLA compatibility, or donors that are familiar but are only sharing one haplotype with the patient, what we call an haploidentical transplant. And then virtually in all the donor recipient, there are also outside of HLA molecular determinants that we call it minor histocompatibility antigens. And we will focus in particular on this second category of donor-recipient disparities. And minor histocompatibility antigens are a broad category of antigens, so they are peptides that are presented in the context of HLA that arise from differences in the amino acidic sequence from virtually any gene in any donor-recipient patient, which are bystander products of evolution, those polymorphisms. And that can be classified based on the fact that they are located in the Y chromosome, so that applies to female-to-male transplants, or autosomal when they are outside of the Y chromosome. And what we know about minor antigens is that 159 have been thoroughly characterized so far across 21 HLA class 1 alleles. But this is just the tip of an iceberg, because if we think about how many exonic variants that give rise to amino acidic differences in any given individual, the number is mind-boggling, because it's more than 25,000. And if we add the additional layer of how many HLA class 1 alleles exist in the population, this number becomes pretty impressive. And to add an additional level of complexity, there is also the ancestry background that kinds of influence both which variants and which HLA class 1 alleles we could have in any given individual and any given donor-recipient pair. So basically, there's a lot that we don't know. And characterizing each minor antigen individually has been a tremendous effort, very not so very cost-effective in the sense that is very intensive from an experimental standpoint. And it's impossible through this kind of methodology to characterize the full antigenic landscape at a scale and for any individual donor recipient pair. So, but this is still sort of the background on which we started this project. And what we were lucky enough to be, it was a time in which the advances in next generation sequencing and single cell sequencing, together with the presence of epitope predictions tool for predicting which peptides could bind any given HLA allele, together with the presence of pipelines that have been motivated, generated for new antigen prediction for solid tumors. All these kind of building blocks positioned us in a place where we could try to approach the minor antigens in the era of precision genomics. So what we did was to develop a computational pipeline to predict minor antigens at a scale. And we did some efforts in validating the antigenicity and the immunogenicity of this pipeline. And then we applied it to a cohort of 220 patients that were transplanted at our institution, so Dana-Farber, from a related sibling donor. We focused on myeloid disease, so AML and MDS. And then we try to see whether the predicted minor antigens had correlations with GVHD on one side, and on the other side, if we could use this platform also to identify potential targets for immunotherapy, because we know that especially for acute myeloid leukemia, finding targets that could be used for immunotherapy is kind of a challenge. So our pipeline starts from whole exome sequencing of donor-recipient pairs, and this is something that is becoming increasingly affordable. So it's within the range also of what we do as clinical testing for routine diagnostic in cancer care. So something that we can really think about implementing in the clinic, hopefully in the near future. What we do is identify non-synonymous patient-specific variants, and at this point, we kind of hone in on the genes that we are interested in. And for this, we developed some filters for application for GVHD or GVL that I will mention later. Got lost in one slide. And the reason why we generated this filter is that minor antigen tissue expression is really crucial to dictate whether minor antigens could serve as for GVL or GVHD, because we have genes that are expressed everywhere that would mediate both GVL and GVHD, genes that are expressed in epithelial tissue that could be targeted just for GVHD. And conversely, those genes that are only expressed in the hematopoietic system are those that could mediate a truly graft versus leukemia effect. So we have to take into consideration really which is the pattern of expression on a gene. And that's the reason for these filters. But when we have decided which filter to use and which genes to put in those filters, what we do is that we generate all the possible 8 to 11 amino acid long peptides that encompass the variant, the polymorphism of interest. And then all these peptides are subjected to HLA class 1 epitope prediction, so that the output is a list of candidate minor antigens for which we know the pattern of expression. So for GVHD, what we used is single-cell RNA-seq dataset, and the reason why we did this and across different organs that are target of GVHD is that compared to bulk sequencing, single-cell allows a precise identification also of resident cell types that are numerically small and thereby might be not precisely captured by bulk sequencing of the tissues. So basically we annotated all the tissue resident cell types, and we retained all the genes that had a count per million more than 5 in at least one of the clusters. And so we compiled this atlas that has a little bit more than 13,000 genes. Some of them are pan-expressed and some are specific to some of the organs. Then for female to male transplant, as I mentioned, there are Y-encoded minor antigens. So there are genes that are in the male-specific region of the Y chromosome that basically the donor T cell of a female donor would never encounter. So there's no deletion at the thymic level of T cells potentially recognizing them. So they are pretty immunogenic. There are 9 genes in this region that are expressed outside of the developmental phases of the testis, and this is an information that we got from GTEx. And we followed exactly the same process as I mentioned before, so generating those 𝐏𝐚𝐅s that we then subject to HLA class 1 epitope prediction. And this is just an idea for any individual HLA allele A, B, or C, which is the distribution of the epitopes coming from any of these genes. And finally, for graft versus leukemia, we follow a different approach because what we want to find is genes that are preferential expression in AML. And we want to target this for immunotherapy. So we really don't want these genes to be expressed anywhere else. So we used a single cell derived molecular classifier to define expression clusters in a huge AML cohort. So it's 451 samples at diagnosis for which there was available RNA expression data. And we found that at least 20,000 genes were expressed across these expression clusters. And then we filtered out whatever was expressed in a non-hematopoietic system. And we were very stringent, and we did 2 rounds using RNA level and protein level data from the GTEx repository. And through this process, basically, we found 259 candidate genes. And this is the expression pattern across the clusters and in the healthy tissues. As you can see, there's a little bit of tolerance for testis antigens, which we know that we can target. And then the question is whether these predicted minor antigens are immunogenic. And to do this, we use the Y minor antigens because they are a little bit easier to handle for experimental purposes. So we unbiasedly took all the predicted epitopes across 6 HLA class 1 alleles. We expanded T cells T cells from female donors against synthetic peptides corresponding to these epitopes, and then we looked at whether we could find or not minor antigen specific T cells. And here we have some results, some representative plots across the different HLA A, B, and C alleles. And then every bar that goes
2 minutes.
Oh, already? Okay, so I need to be faster. This is what we get in terms of validation. And then the next question is whether this is associated with outcomes. And we found that the load of minor antigens was associated with GVHD in univariate and multivariable analysis, and also with chronic GVHD in female to male and transplant. And lung GVHD was particularly relevant because it's one of the most morbid conditions. And again, lung-specific minor antigens were associated with chronic GVHD in univariate and multivariable modeling. And finally, we looked at GVL minor antigens. For this, we looked at a specific category of patients that were the ones that didn't relapse and didn't have GVHD, so the GRFS patients. And we found a recurrent set of SNPs in these patients, and this is how many per patient. And we looked at, when we stratified the entire cohort by this group of GRFS antigens, they were protective against relapse. And we validated this finding also in another external cohort from Spain. So I hope that I convinced you that minor antigen prediction is feasible and scalable and could help GVHD prognostication to tailor risk mitigation strategies for high-risk patients, and also to identify targets for immunotherapy. And basically, this is also the beginning of when you build a prototype, you want to do something bigger and more beautiful. So we're working on expanding the classes of alloreactive targets to predict and to apply this in a contemporary moving field with PTs, with new GVHD prophylaxis, with the ultimate goal of finding a universal risk score for all the patients. And finally, the idea is to target. I had to have some car to feel part of the group. So the idea is to target GVL minor antigens with TCR-like CARs. And with that, I would just like to thank everyone that has contributed to this work and you for your the attention. Thank you. And now I have to find how to stop sharing.
So thank you for this really exciting presentation. I just forgot to mention who we are, the jurors. So I'm Alessandra Ferrajoli from MD Anderson in Houston in the leukemia department. I've been at MD Anderson for 27 years, of which 25 dedicated to the leukemia department. And then Marco Ruella is from the University of Pennsylvania. He's an expert in CAR-T. I think you are an assistant professor, is that right? Or you're associate now? Is associate.
You're on mute.
I can read the lips. And right at
Very
the same point in career, there is Dr. Paolo Strati, that is an associate professor in the Department of Lymphoma here at MD Anderson, and is also an expert in CAR-T as well.
I just wanted to add, I got the Premio Campese in 2016, I think. So
correct.
I want, you know,
I was there, Marco.
I know, but I was very happy to sort of be called back to be a juror. It's kind of nice to see things, you know, that continue. So. And
Dr. Strati was also a finalist a few years ago.
But I didn't make it. Did you win over me, Marco, the year? It was either
no,
2015
we
or 2004. Yeah.
I don't think it was
maybe
the
it was
same
2000.
year.
Now it was 2014 or 13.
Yeah, I think you were 1 or 2 years before. Now, going to the very exciting presentation of Nicoletta, I would like to ask a question because I'm really intrigued. So when you try to scale this at the individual patient, do you need to have the blast from that individual patient, or your large, you know, trial, you use the database from the Stop AML? Was that enough to create a variety of the, what can I say, the characteristics, the antigens in the blast to predict your model?
The pipeline is built in a way that we can input both DNA and RNA. So basically, if we have the opportunity to have the blast of the patient, we can provide that level of information that basically will get us to evaluate whether in that specific minor antigens are expressed in the blasts. So at the population level, we are pretty confident about the expression. Obviously, if we add that level of information, which will not be available for everyone, obviously that would be a plus, which is the same thing that is done also for neoantigens in solid tumors, for example. So whenever that is available, it's always better. But yeah. Especially in leukemia, it's kind of more difficult because RNA, it's not something that is archival, and there's no FFP that you can use. So especially when you are a third level center, you might have transplant patients that were treated and diagnosed outside of there, and getting that level of information can be difficult.
Correct. That's the challenge, and the challenge also of the change in the leukemia feature with each relapse that can be particularly challenging if you don't have different type of materials.
Exactly. In fact, one of the things that we are working on is also to characterize leukemias at relapse. And now that we have this instrument, this tool, basically what we want to see is at different relapses and compared to when the transplant was performed, we can see how these antigens fare. So if they are depleted, kept, and which ones are the one that we have to bet on.
Then very quickly, before I give a few seconds to my colleague, do you see an application in autoimmune or rheumatological disease outside the hematological field?
So that's a very interesting question. So the first thing that I've started thinking is solid organ transplantation. So that is something that I'm interested in doing. But for autoimmunity, the problem is that what we have is genes that are overexpressed. So there's something that can be done on that. There are some studies that have looked at, for example, cardiotoxicity, and looked at what was expressed at the level of the thymus, and focusing on what was not expressed there, basically, to model what could be the target of autoimmunity.
Thank you. Now I leave it to Paolo and Marco for questions.
So, maybe, go ahead.
No, go ahead, Paolo.
Okay, I'll go first. First of all, congratulations, Dr. Cherry. That's amazing work, quite big bulk of work performed in a relatively small amount of time. So really congratulations. I have two general questions. So you alluded to, toward the end of your presentation, to the potential clinical application of your findings with immunotherapy targeting these antigens in the post-transplant setting. Could you comment a little bit more about ongoing efforts and how you plan to address the barrier of immunosuppression that would be given, of course, in the post-transplant setting?
So yes, what we are currently doing, and we got lucky enough to get ASH funding for that, is to basically find, so focusing on these GVL minor antigens, look at first AML cell lines by mass spectrometry to evaluate which one we can find proof of presentation in the context of HLA. So for those that are validated at that level, what we are using is a screening with a nanobody library to find antibodies that recognize specifically the peptide in the context of the HLA. And at that point, we're doing screening right now for 3 candidate minor antigens. Those could be used in the backbone of a CAR, and the idea is to use the donor T cells, which we can for which we can delete the endogenous TCR, so we can get rid, hopefully, of GVHD, and then use it for post-transplant immunotherapy. So it would be cells outside of the individual. So immune suppression, well, will depend on when is the window of relapse for the patient.
Mm-hmm.
Now let's move to Marco, because
Yes.
we're a little late.
Yes,
Yeah,
yes,
we're
yes.
running out of time, so I'm going to ask a different question. I want to ask about your career development. What are the next steps for you in your career?
Applying for assistant professorship.
Okay, so you're going to be in the job market for assistant professorship and in the tenure track with the lab. Do you want to see patients as well?
Yes, that's something that I'm reasoning about, with all the inherent difficulties of being Italian and trained in Italy and not wanting to go back to residency here. So
well, we can talk about
actively.
that. We can
Yeah.
talk
I
about,
know.
we can
I
talk
think
about
all
that
the
offline.
Yeah,
three
some of us have done it.
3 of us
Exactly.
offline and follow different paths, and
Yeah.
we all end up there. Great.
Yeah,
Now-
the problem is also the state differences.
Right. Well, we
Yeah,
live
very
in the
true.
state of Texas, let me tell you. We will move now to the next presenter, that is, I'm going in alphabetic order. So the next presenter is going to be Sara Piccinelli. And Sara Piccinelli will be discussing for us the overcoming CAR T-cell resistance in acute myeloid leukemia. So Sara, please go ahead.
Yes, I'm sharing my screen. Okay. So can you see my presentation?
Yes, we can.
Okay, perfect. So I'm going to discuss, this is the project that I've been working on during my postdoc in Dana-Farber in the Romi lab. So basically what we tried to do, it was like to find a new approach to treat acute myeloid leukemia. And in particular, the project is about combination therapy in which we use both CAR T cells and menin inhibitors. So acute myeloid leukemia is still an unmet clinical need. Indeed, we didn't have like any significant improvements in the past 40 years, like you can see from this graph. And also CAR T cells is actually really improving the outcome on some of different hematological malignancies. For acute myeloid leukemia, they are not still good or effective. So one thing that a novelty that it happened recently, it was the FDA approve of menin inhibitors. Menin inhibitors are a class of small molecules that basically inhibit this molecule called menin. And mannin is important in the process of leukemogenesis, and especially is overactivated in two kinds of acute myeloid leukemia with two kinds of mutation. One is NPM1, and the other one is KMT2A rearranged. So the drug has been approved for KMT2A at the moment, but it's also effective in an NPM1 mutant clone. So the activity of the drug basically is to inhibit the activity of mannin, and this brings like to differentiate the leukemia cells, like to bring them back to a pre-differentiation and like stem cell state. And so mannin now have been, many inhibitors have been tried in different clinical trials. This is one of the first one that we published in Nature 2 years ago. And so we can see that we tested in both the mutation, KMT2A and NPM. Efficacy was like 20% of complete remission. So here we can see like mannin is decreasing the expression, and we see an increase in the differentiation like molecules. The problem of this therapy and that mannin acquires mutations. So this is the reason why, even if it's really, the efficiency is very high, in the end, patients relapse. And the relapse is also like pretty quick. So in this study, after a couple of cycles of therapy, 40% of patients acquire mutation. So you can see here on the right, like after 2 cycles of therapy, they can acquire one or multiple mutation in mannin. So mannin is not more like targeted by mannin inhibitors. So what we thought is like, why we don't combine like CAR T cells that they are like partially effective in acute myeloid leukemia with mannin. They're also partially effective in order to see if we can see like an improvement in the treatment of the blast. And also, the problem of mannin, we say the mannin inhibitor, is that the mannin acquire mutation, but CAR T cells should be effective also against the leukemia clones that carry the mutation. So we try to demonstrate that our hypothesis work. So the first things we did is to test the T cells with mannin to see if T cells are affected by the treatment with mannin. So you can see here we test T cells in different activation status, both for like cells growth and functionality. So we treated them with a different concentration of the unit. That is like the most classical mannin inhibitor that has been used in the clinic. We can see that after 5 days of incubation, we don't see difference in the expansion and proliferation in the cells. And also, we don't see difference in the functionality in terms of CD107 expression, interferon gamma, and TNF-alpha. After that, we demonstrate that T cells, they can remain active in the presence of mannin. We had to decide like which CAR we want to use for this project. So we decide to use 2 different CAR constructs. So one for the NPM1 mutant clones and one for the KMT2A. So for 1PM mutant clones, we use a CAR that we have been designing in our lab a few years ago. It's a TCR-like CAR who actually bind the complex like class 1 A2 with the AQ that is the new epitope who is produced by the CAR, the NPM1 mutant. And for KMT2A range clones, we decide to use a CAR, the CD70 CAR, because CD70 is actually a surface antigen that is really quite present in like 40–90% of leukemia blasts, but it's not present, it's not expressed in stem cells. So the CAR basically express the CD27 ectodomain that binds CD70 on the leukemic cells. So starting with the combination with the NPM1 CAR plus mannin, So I start working on in vitro data. So I use mostly those two cell lines. One is MLOC3 because it's the only cell line that we have that carry the mutation for NPM. And we have another cell lines that is the same cell lines in which we introduced the resistance for many. Basically, one of the mutations that have been detected from patients. So how you can see when we treat the cell lines with different doses of removaline, OC3, they are pretty sensitive with increasing the dose, but the cell lines that is resistant, it start to be affected only for a really high dose. They actually, they're not the dose that are used in the clinic. So we tested the capacity of the CAR to kill and, to be functional against the cell lines and to kill them. So we can see here on the right that the CAR is functional against both the cell lines with or without the mutation, and also the CAR is able to kill the cell lines, the wild type and the one with the mutation. After that, we want to see the effect of the combination with the CAR and manning. So basically, we repeated the killing, but we pre-incubated the cell lines with a small dose of manning for 4 days, and then we added the CAR and incubated the CAR for 24 hours. So you can see on OC3, this is the effect of the CAR by themselves, and this is the CAR plus manning. And with the mutant variant, you can see that the CAR still kill, but there is not this big difference like in the other cell lines, because the mutation doesn't allow many to be efficient. And so because we had these good results in vitro, we decided to move in vivo. And this is the mouse model with the sign. So we use a PDX with carrying the NPM1 mutation. After the engraftment, we inject 50 million of CAR T cells, actually 50% of transduction, so let's say 8 million. At the same time, we start the co-treatment with 𝐕𝑖𝑡𝑜. It was like a chow for the mouse for 1 month, 30 days. And we did this because we want to see the effect in combination, but also we wanted to stop the treatment at a certain point in order to see if the mice were cured. I also like to mimic relapse and see if the CAR were able to treat the relapse. So how you can see here, this is the leukemia burden in the peripheral blood. We have the CAR T cell. They are able to control for a while the disease. 2 mice, up to 4 we lost at the beginning, and then the other 2 mice we lost here when at day 70 we had the relapse. The mice that were only treated with manning in blue, so they had a really good response, but when we stopped manning, we saw the relapse and we lost all the mice. But in red, we have the mice who received the combination, so they also have a really deep response at the beginning, and they had a little bit of relapse. They increase here at the 70, here is not really visible, but then the CAR, they were able to control the relapse, and after 7 months, they were still alive, and then we sucked them and analyzed the organ. So you can see here the survivor. We lost 1 mouse because of extramedullary disease, but for the rest, the other 3 mice, they were alive for 7 months, and when analyzed the organs in the end, they are leukemia free. Another interesting thing, we were able to see what's happened to our CAR T cells. So we monitored the mice every 10 days in the peripheral blood. So we can see that the mice only with the CAR, they had this big expansion of CD3 at the beginning, and they were still trying to fight the leukemia. And in the end, we lost the mice. But the mice with the combination, they had an expansion of CAR T cells, but probably the control of the leukemia with the double therapy were be able to not let them expand too much. But when we had the relapse, the cells were still there, and we had this huge expansion at the 70, and then they were able to control the disease. So this expansion, you can see here for the GFP positive, were mostly CAR. So some mice that were like 90% only CAR. I remember at the beginning we injected them, they were like 50% transduction. And the interesting thing is that while at the beginning we had like the expansion was mostly CD8, at this point, we see that these cells, they were mostly CD4. So we had the huge expansion of CD4 CAR T cell. They were able to control the relapse. We are still working on the CAR T memory phenotype because we are trying to demonstrate that the combination, maybe because of the timing, maybe for other mechanisms, is able to make the cell like to switch from a CD8 to a CD4 phenotype and maybe memory. This data are still ongoing. For the CD70 CAR, we did a similar approach. So we start with in vitro data. We use like those other cell lines and 𝑉411 who carry the KMT2A range. And we also have the clone with the mutation. Also here you can see how the cell lines is sensitive to manning, and the one with the mutation is sensitive only for really high dose. We check functionality of the CAR and it works really well, and we check the killing. You can see also, I mean, it's a really potent CAR, different ET ratio, works really well. And then we did the same approach. We pretreat the cell lines with manning, a really low dose because those cells are pretty sensitive. And then we add the CAR after 4 days of incubation, and you can see how the CAR works well. But with adding manning, I mean, the effect is much higher, especially for really low ET ratio. And here you can see how doing the same with the cell lines that carry the mutation, we don't have any effect about manning, but the CAR still works. So the CAR should still be able to control the clone with carrying the mutation. And here we also use a similar approach. We wanted to complicate it a little bit. So we use
2 minutes.
we inject the, that was okay, I will go quickly. So we'll inject the mice with 𝑉411. We use a mixture, 80% wild type, 20% with the mutation, because we want a real like relapse. We inject the CAR T cell, 5 million, and we treat the mice for 2 months with Revunit. You can see here the BLI, so leukemia burden. So CAR T cells in red control a little bit, but they don't cure the mice. Many control a little bit better, but then start to relapse, probably because of the mutant clone. But you can see here that the combination, the mice were basically from the beginning, they clear leukemia really, really well. So here we suck the mice at day 90 to analyze the organs. And they are all in remission. And here you can see also the expansion of the CAR. So there is a huge expansion in a CAR T cell because they were trying to control the leukemia. And also here for the combination, after the first expansion, we saw that basically CAR, they don't have the target anymore. So they are really detectable in the peripheral blood. And here, so the expansion was mostly CD8, but I have the data on the organ that they show that at the end also there is a predominance in CD4. And we also are like doing now the genotyping to see how, I mean, the phenotype of the relapse and the leukemia cells that relapsed. And quickly, we tried to check if apart from the combination therapy, there was some kind of mechanism that could kind of explain this potent effect. So we thought about what menin does, I mean, about the expression of the target. So we check here in flow cytometry, but we have some mRNA sequencing data ongoing, the expression of class 1 on the target cell for NTM, and we see that even with a short dose after 5 days of incubation, we have an increase in class 2 molecules, and the same happened for CD70 and those other cell lines. So this could be another mechanism. I mean, menin inhibitor upregulate the target, and so this can lead to an increase on the effect. And another thing, we wanted to see if menin were able to make the cells more pro-apoptotic, like to make them cells easier to die. So we use the BH3 profiling assay. You can see that after 24 hours incubation, all the cell lines, apart this one with the mutation, I think it's kind of resistant. It's maybe 24 hours not enough, but all the cells, they kind of are like an pro-apoptotic priming. And so, yeah. So now the future plans for this project, we are trying to define a clinical trial. This is not the real clinical trial, so we are thinking about this. We are presenting data to ASH, and we are going to talk with other centers in order to see if someone is interested to start a trial with one of the two car. And then, yeah, there is also this other project to create a tandem CAR in order to increase, especially because NPM is a little bit less, the affinity of NPM1 is a little bit less than CD70. Try to combine the two in order to have even a strong CAR and have also a deeper effect. And yeah, thank you all for listening to me. And this is my lab, so I thank for my lab member and collaborator. Thank you.
So thank you for this exciting presentation, and definitely, you know, these two types of leukemia are the one where, especially the KMT2A rearrange, we have great difficulty in controlling clinically. I have a question, but I'll let my two other reviewer go first, so I don't take all the time.
Yeah.
No, Alessandra, go ahead, please. You are the leukemia doc here.
Okay, so my question, and it's a little bit, you indicated that on your last slide, is looking at clinical application. When do you think is the best timing for this type of approach?
When they are in MRD. Because I try an experiment in which actually it was not something that we expected, because we try another experiment with the PDX, and in the end, when I inject the mice, the leukemia burden was really high. I didn't want that, but I mean, we arrived at the point. And yeah, I mean, even if we did the mice with menin alone, they did well for a while, but the combination therapy, I think the leukemia burden was so high that we had toxicity from the CAR. So I will suggest, I mean, at least to start with patient MRD or really small amount of leukemia at the beginning to see. Also because both, I think no one of the two CAR have been tried in patients yet, at least not the NPM. So we will have to start with safety, like I mean,
CD70 CAR is in clinical trial, but not in combination with the menin inhibitor. So you will be having a patient on menin inhibitors, either, you know, any of them, and then add the CAR T plus menin, and at that time you would be stopping the menin, considering that the menin with the CAR T can take over, or you can consider even a double administration with this.
I won't stop menin.
Okay.
I will give them. I mean, this is resign shoulder.
Right. Just.
But I will start together, and I will then stop menin.
Well, then Marco and Paolo, please.
Yeah, I can get started. So very nice work. So we've done some work on epigenetic modulation and CAR T-cell, and actually we were thinking about also doing menin inhibitor and CAR, but since you're doing, we're not going to do it, okay?
But there are several menin inhibitors you can use.
Yeah, there is 𝑉𝑖𝑡𝑜, there are a bunch,
Enzo.
No, but just to say that I think it's a clever approach, very pragmatical, right? Because you can use, in theory, FDA-approved agents. In theory, again, as Alessandra was mentioning, I think 𝑉𝑖𝑡𝑜 should be approved before the end of the year. Revumenib should also be approved for NPM1 by the end of the year, and there is a bunch of other ones that are in the pipeline. Nevertheless, when we were doing experiment with epigenetic modulator, is always the question, is the fact that we're seeing, you know, mostly on the tumor, mostly on the T cell, in both. So I noticed that in the in vitro work, you were pre-incubating the tumor and then adding the CAR, if I understood correctly. And then when you switch to the in vivo, you're giving actually the drug after the CAR infusion. So have you seen in vitro that if you keep the drug in the culture, it gets maybe toxic to the T cell? What is the effect of the drug on the T cell?
It was my first slide. So I did the incubation for like 5-6 days with the drug and T cells, and they don't affect, proliferation, they don't affect the activity of T cells. Also, I know there are people in Sloan Kettering, they're studying the effect of menin on T cells, and it seems it can be like good and not bad. It seems that in some way they modulate T cells in order to be more activated or more effective. So I don't know. I was talking with Scott Armstrong because they are our collaborator, and I know there are people interested in this, and they didn't see in their data, in patient data, any effect on T cells. They know there is like some effect on like granule size, but T cells, they don't seem to be affected. Also in vivo, in mice, you can see that, T cells are really effective even after days that mice are treated with the drug. So, yeah.
Okay, one quick question for Dr. Strati.
Yeah, it's more a suggestion. I'm gonna quickly comment on the immune cell subtype that nobody's mentioning today, which are monocytes and macrophages. As you may know, in preclinical models of CAR-T and now in translational models, it's very evident that monocytes and macrophages not only mediate CAR-T cell toxicity, but also function. If you give clodronate and you completely deplete macrophages, CAR-T cells actually meaningfully decrease their activity. And again, I'm not a leukemia doctor, but I'm pretty confident that menin inhibitors do have some, I used to 10 years ago, but menin inhibitors should have some impact on monocytes and macrophages. So my suggestion would be, and maybe you're already doing that, to also have co-cultural studies where you have, both monocytes, macrophages, and CAR-T cells, and making sure that menin inhibitors may not indirectly impact negatively the activity of CAR-T cells. And maybe you're already doing that.
Well, that's very pertinent because in clinic, when we see the differentiation syndrome, some of it is probably driven by the monocyte and macrophages. So that's very important. Thank you again. Thank you, Sarah, for this beautiful and exciting presentation. Now we will first thank you, Dr. Rotiroti, for her patience, because it's not easy to be the last one, but we'll proceed with the last presentation from Maria Caterina Rotiroti, and she will be discussing chimeric antigen receptor CAR-T cell therapy for hematological malignancies.
Thank you for the introduction.
Maria Caterina, go ahead. Yes.
I hope you can hear and see my screen. So in the context of CAR-T cell therapies for hematological malignancies, the main focus of my postdoctoral work was to enhance the activity of CARs towards lower antigen densities. It is now very well known that CAR-T cells require a high antigen density for activation, and their level of activation is actually proportional with the amount of antigen used for stimulation. During my PhD, I actually showed this phenomenon in the characterization of anti-CD123 CAR for the treatment of AML, where we can see how in response of target cells expressing different levels of the target antigen, we have a level of activation, in this case measured as cytokine production, that increases with the level of density of CD123. And this is a pretty general phenomenon that holds true for CARs with different specificities. And actually, in the same year, my current mentor published similar results for the targeting of the anaplastic lymphoma kinase, where we can see how in response of higher levels of antigen density, we have increased activation of the CAR-T cells. And despite this is a feature that can provide a therapeutic window for the targeting of tumor-associated antigens whose expression is also present on healthy tissue, at the same time, this opens the way for tumor resistance through antigen downregulation. And unfortunately, this is a phenomenon that has been described in clinic. So we can see here how patients treated with the CD22 CAR experience CD22 downregulation and relapse as compared to pre-treatment. And in a different disease context, being multiple myeloma and BCMA targeting, we can see how patients who initially respond to treatment have a lower level of BCMA, while the level tends to go back up once the treatment stops working. And patients who never experienced a benefit from the treatment do not really see this downregulation of the antigen. So in my current lab, we have recently shown how, at least generally speaking, the propagation of the T cell signal, of the CAR signaling, relies on the same signaling molecules as TCR. Briefly, the SAR kinase LCK, after antigen engagement, phosphorylates the CD3 items, forming docking sites for the 𝐸𝑒𝑑 tyrosine kinase that then phosphorylates latent 𝐙LP76 to form this signalosome that recruits additional signaling molecules, including PLC gamma, leading to the final T cell activation. And here we can see how when we knock out one of these main players, we basically abrogate the capacity of a CD19 CAR to activate in response of antigen stimulation. So despite relying on the same signaling components of a natural T cell, while a natural TCR is able to respond to even 1 to 10 molecules, peptide molecules once presented in MHC, the antigen activation threshold for CAR is significantly higher. And different groups have attributed this to an inefficient recruitment of proximal signaling molecule to the immune synapse and reduced phosphorylation. For example, here we can see when the same T cell specific for CMV is engineered with a ROR1 CAR, there is much less 𝐸𝑒𝑑 recruitment and phosphorylation following stimulation. And similarly, another group has shown by looking at phosphorylation events how the TCR stimulation show a stronger phosphorylation and therefore activation profile of these signaling molecules. Work from our lab has shown that minimal architectural changes have the power to modulate this antigen sensitivity of CARs. So here we have the schematic of 2 very well-known CAR approved in clinic, 𝐂𝐚𝐨 on the left and 𝑉𝑖𝑡𝑜 on the right. And we know from preclinical work and clinical experience that 𝐂𝐚𝐨 is well known for its longer persistence, while 𝑉𝑖𝑡𝑜 for being more sensitive to low antigen densities. And this was attributed to the stronger signaling component CD28. However, here we can see how these 2 CARs also differ for the hinge transmembrane region. And by simply providing a 𝐂𝐚𝐨 CAR with the CD28 hinge transmembrane domain, we are able to restore the sensitivity of this CAR, matching the efficacy of a CD28 co-stimulated CAR. So here we can see how in blue, in this model of low antigen density leukemia, the CAR, the original 𝐂𝐚𝐨 design has minimal activity, but once we provide the 28 hinge transmembrane domain, is able to match in efficacy a CD28 co-stimulated CAR. So when I joined the Metzner lab, I wanted to study the signaling dynamics between these 2 CARs, and we performed the phospho-mass spectrometry after stimulation. So we can see here how the CD20, by looking at phosphorylation events, the CD28 hinge transmembrane domain is associated with the faster and stronger phosphorylation of both proximal and distal signaling molecules. So I tested the hypothesis that maybe providing more of these signaling molecules would compensate for limited CAR sensitivity and low antigen density. I started overexpressing these 5 molecules together with the CD19 CAR, and we can see how the overexpression of 𝐸𝑒𝑑 and 𝐙LP76 actually resulted in enhanced activation. However, this was in response of an 𝐴𝑚6 cell line, which is a BLL cell line expressing relatively high levels of CD19, which is about 20,000 molecules of CD19. And when we challenged these same cells against our engineered 𝐴𝑚6 cell line expressing about 1,000 molecules of CD19, we didn't actually see any enhancement. We were still encouraged by the previous results, and we wondered if maybe tethering these these cytosolic molecules to the membrane could further improve their efficacy. So we started providing a transmembrane domain to 𝐶𝑎𝑚70 and 𝐶𝑠LIP76. Unfortunately, we were not able to overexpress a membrane-tethered version of 𝐶𝑎𝑚70, but we were successful in engineering a membrane-tethered version of 𝐶𝑎𝑚76. And this maneuver allowed us not only to enhance the activation in response of high antigen density, but most importantly, to enhance the activation in response of low antigen densities with also improved killing capacity. And when we challenged the CD19 CAR with and without membrane-tethered 𝐶𝑎𝑚76 with an array of NAM6 cell line expressing different levels of CD19, we can really see how we are able to lower the sensitivity, to enhance the sensitivity of the CAR T cells. So we confirmed these results in vivo using the CD19 low leukemia model, where we can see how membrane-tethered 𝐶𝑎𝑚76 confer superior tumor control as compared to the CD19 CAR wild type, or with the overexpression of 𝐶𝑎𝑚76 or 𝐶𝑎𝑚70. And I have mentioned before that CD22 downregulation is actually an important problem in clinic, and the same group who published about this phenomenon actually shows how the CD22 CAR used in the study, while being able to control NAM6 cell line expressing regular antigen density, which is about 4,000 molecules here in green of CD22, when challenged with an NAM6 cell line that has been engineered just to express half of the molecules actually fail. So we took the same CAR, the same CD22 low leukemia model, and we treated the mice with CD22 CAR with membrane-tethered 𝐶𝑎𝑚76, and this was able to control tumor for a long time, and was associated, at least at the initial time points, with the superior expansion of the CAR T cells, both in the bone marrow and the spleen of the treated mice. We wanted to do the same with BCMA, so we took this multiple myeloma cell line that expresses less than 2,000 molecules of BCMA on the surface, and even in this context, we saw improved efficacy when we provided the membrane-tethered 𝐶𝑎𝑚76. As also important check, we wanted to confirm that by providing this signaling boost, we were not going to affect the persistence of a 4-1BB co-stimulated CAR. So we treated mice that have been engrafted with wild-type NAM6, so regular antigen density, and used curative T-cell doses to match efficacy between the CAR with or without membrane-tethered 𝐶𝑎𝑚76, as seen from the tumor control graph. And then we euthanized the mice 21 days after treatment, and we found that we had a similar level of CAR T cells in the spleen, so membrane-tethered 𝐶𝑎𝑚76 didn't affect the persistence of a 4-1BB co-stimulated CAR. Obviously, when considering a strategy to enhance CAR sensitivity, it's extremely important to consider the antigen that we are targeting, because this carries the risk of on-target of tumor toxicity. And actually, as a proof of concept that this could happen by applying a membrane-tethered 𝐶𝑎𝑚76, we took advantage of a toxicity model that was previously published, and we targeted ROR1 with the CAR that is cross-reactive between human and murine antigen. So we engrafted NSG mice with NAM6 expressing ROR1, but ROR1 is present at low level on healthy tissue of the mice, and especially on the lungs. So when we look at tumor control, we can see that both CAR T-cell treatment resulted in tumor control, but the mice that received the membrane-tethered 𝐶𝑎𝑚76 experienced significant toxicity that we measured as tumor as weight change, and we had to euthanize them within a week from the treatment. Finally, we wanted also to understand the molecular mechanism by which membrane-tethered 𝐶𝐶76 was enhancing the activity of the CARs. 𝐶𝐶76 is quite a big protein interacting with several other partners here depicted. So we took advantage of point mutations and deletions that have already been described in literature to prevent these interactions, to figure out if one of these domains was more important than the others. And it turned out that mutations preventing the recruitment and finally actually the engagement of PLC gamma completely abrogated the enhancement provided by overexpressing a membrane-tethered 𝐶𝐶76. As other experiment we performed, we wanted to compare our platform to other modifications that have recently published as a way to enhance CAR activity by acting on the signaling of the CARs. So we used the same CD22 low leukemia model previously described, where the original CAR is able to control the tumor for a certain time, but then the tumor relapses, while membrane-tethered 𝐶𝐶76 is able to control the tumor for a long time. And without going into the details of all the other constructs that we tried, we can see that while some of these constructs were able to increase the efficacy of the CD22 CAR as compared to the original CAR here in black, membrane-tethered 𝐶𝐶76 was outperformed these approaches. So in conclusion, we have shown how overexpressing proximal signaling molecules has the potential to enhance CAR activity, and particularly the genetic engineering of 𝐶𝐶76, providing it to the membrane, is able to lower the sensitivity of CARs, enhance the sensitivity of CARs, and the advantage is that it's a platform that can potentially be applied together with other CARs already designed, so without modifying the backbone of the CAR itself, obviously with particular attention of the target antigen, or eventually integrating safety switches. And what am currently interested into is also understanding if this approach could be used to enhance, for example, the efficacy of low-affinity TCRs, and as a signaling aficionado, also understanding if there is any difference in the way membrane-tethered 𝐶𝐶76 affects CD4 and CD8 CARs. So this is my last slide, which is the acknowledgement of all the members of my lab, which is currently actually much bigger here in Boston, and our collaborators and funding, and thank you for your attention.
All right, nice job.
So thank you very much for your great presentation, really exciting data on even improving something that already works quite well, like the CD19, CD20 CAR, anti-BCMA CAR. So on that line, which are the diseases that you feel this approach could have the highest impact? For example, I'm asking about chronic lymphocytic leukemia, where we know the activity of the CAR is there, but not as good as the one that we see in ALL or lymphomas.
That's a great question. I think the disease, so we need to consider there are several reasons why CAR fails, and antigen remodeling has been reported in BCMA, CD22, and also in a clinical trial with the dual targeting of CD19 and CD22. Probably for CLL, the mechanism of resistance are not necessarily due to antigen remodeling. Therefore, applying a boost where unfortunately, there is, besides the on-target off-tumor effect, there could be also exacerbation of cytokine release syndrome, would not be ideal. So I envision this to be applied to relapse with low antigen densities in leukemia, acute leukemia.
Okay, now Dr. Ruella and Dr. Strati, since you are CAR-T aficionados.
Maybe I'll go first this time. So first I'll ask the question that Marco asked to one of the prior presenters. What are your career plans?
I also would like to have my own research group back to Italy. Okay. So I started applying to starting grant, European and Italian ones. But
that doesn't mean they will not maintain your ties to the US. You are creating right now a network and collaborators. I also want to make a quick comment, and I'm sure you're aware of this. There are already out there TCR T cells that have shown in preclinical models of being quite active in either CD19 negative or CD low lymphoma models. So I guess you could apply the same CD22. So that's definitely an area along with CAR T cells that you may want to eventually invest into moving forward. Just an advice.
Thank you for the suggestion.
So nice work, Maria Caterina. So the enhancement was also present when you would use, and I haven't read your paper, sorry yet. It just came out recently.
It came out yesterday. So.
I know. I was aware of it. I was aware of the paper because I think Robbie posted it somewhere in LinkedIn. And I really like this approach. Of course, I knew about the data from conferences, but so this system would work also if you have sort of your optimized 4-1BB with the CD28 transmembrane and hinge.
Yes, actually, many of the in vivo experiments utilize the CD28 hinge transmembrane domain. We haven't, so the current design for a membrane-tethered SLP76 has a CD8 hinge transmembrane domain, and we have tried the CD28 as well. They both work well, but the CD8, no matter which transmembrane has the CAR, allows us to achieve a higher expression on the surface, so that's why we went with the CD8 hinge transmembrane. But we didn't find any correlation between the hinge transmembrane of the CAR and the hinge transmembrane of SLP76.
And so this is also with CD19. So if you have a normal regular NAM6 with normal expression, is this system also working?
Yeah, in order to see that, we have to do a stress test, which again, we don't know the overall clinical significance of a stress test, because we all know that Kymriah fails in a stress test, but has excellent activity in vivo. So it does work in, we do see differences in stress test by comparing a regular CD19 CAR and regular antigen density with membrane-tethered SLP76. We mainly focused on low antigen densities, but if we stress the system, we are able to see it even in CARs that work very well.
Thank you. So at this point, I would say that all the three of you had presented outstanding work. The applications was very competitive this year. There were many outstanding projects that were presented. So congratulations again. And then just from a personal perspective, it's very nice to see such young female scientists having a really great career in front of you. And this is also part of ISSNAF, where all of us want to try to favor your career as much as we can. So please reach out for any advice. We look forward for the final event that will take in Washington, D.C. Unfortunately, I'm on inpatient rounds that day, so I will not be able to attend in person, but I think for sure my other reviewers, Dr. Strati, and I think possibly Dr. Ruella too, may be able to be there in person and interact with you and give more direct advice. So congratulations again for a job well done.